So, hi everyone, and welcome to the 19th TIA Centre seminar of the academic year. So, my name is Adam Shepherd, and I'm a postdoc here at the TIA Centre. So, today I'm delighted to introduce Dr. Drew Williamson. Drew Williamson completed a BA in mathematics at Oberlin College, an MD at Case Western Reserve University, residency in anatomic pathology at Brigham and Women's Hospital, and fellowships in molecular genetic pathology and clinical informatics at Mass General Brigham. This training was followed by postdoctoral research in the lab of Faisal Mahmood, Faisal Mahmood. A board-certified pathologist, Drew's research focused on the applications of deep learning-based methods to pathology data, from histology images to genomics, to most recently natural language. As of January 2024, he is an assistant professor in the Department of Pathology at Emory University. The title of Drew's talk is Histologic Features, Everything Old is New Again. So thanks again, Drew, for joining us. We're really excited to hear your talk. So, and whenever you're ready, please feel free to start.
Sure. Thank you so much for the introduction and the invitation. And just make sure everything still look good.
Yes, we can see that. Okay.
Okay, great. Let me just move the Zoom over. Okay, great. So good morning, good afternoon, good evening, I guess, depending on where you are in the world. Yeah, so my name is Drew, and I'm a pathologist. So this, I guess, may be interesting, maybe boring to you all, depending on what sort of background you're coming from. If you're coming from machine learning side, I probably won't delve too far into the technical details that may interest you most, but talk about pathology details that interest me. And specifically, I'm going to be talking about histologic features. And by histologic features, I mean these like characteristics of an image that a histology image that describe what you see in them. And so let me try to advance my slides and make sure that works. Okay, great. And so like a very common thing that happens to you when you're going through pathology residency is, especially at the beginning, you like plop down a slide on the microscope and you're sitting across the microscope from an attending pathologist. And they'll just ask you to describe what you see on the slide. And so you got a slide like this. This is a core needle biopsy of a patient's breast mass. And, you know, when you're a medical student or like a first-year pathology resident, you look at this and you're like, it's pink and there's purple bits and I see some cell nuclei here and they're forming circles and maybe some ellipses and that sort of thing. And the attending's not very happy with that histologic description because you're not using the jargon of pathology and you're not really describing the things that they see and the way their brain works or has been programmed to work over years of pathology experience. And this process of developing a description of a histology image is kind of central to how you learn to practice pathology. And it's also got a very long history. And so this is an English translation of the first description of cancer under the microscope. It was originally published in German in 1838, translated to English in 1840. And the original was by a Dr. Müller. And so if you're familiar with the Müllerian system from embryology or from pathology, the Müllerian system is named for this Dr. Müller. And so up until 1838, pathology and the study of cancer was really like a macroscopic discipline. So you look at the sort of gross, as we say in pathology, version of cancer. And so you have these different, at this time, terms that describe cancer only from this macroscopic level. And Dr. Müller was the first person to throw this under the microscope and see and sort of describe what he saw under there. And so this predates H&E, the stain that is kind of the workhorse for pathology nowadays. But nonetheless, you have some interesting histologic features that Dr. Müller describes. And so, you know, cells he recognized are kind of form the basis of cancer. And you know, there's some interesting descriptions that aren't using maybe the jargon of pathology that pathologists expect in 2024, but nonetheless sort of describe what he's seeing there. So these minute globular cells, who knows what that means, and deposited an extraordinary number in the meshes of a fibrous texture. I think, like, you know, that kind of looks kind of similar maybe to this. Maybe these cells are deposited in the Norris number and the fibrous texture back here. And so, from the very beginning, these histologic features that we as a community of people looking at cancer are extracting are in the form of this natural language or some jargon like that. And some interesting descriptions like here, like a cell appears to contain corpuscles in its interior. So I'm not sure if that's like the nuclei of the cells that he was seeing or nucleoli or what he was looking at. But what was interesting to me looking at this, the first paper looking at cancer under the microscope, is that essentially instantaneously we have both histologic features in natural language and in the form of numbers, so like quantifying these histologic features. And so, he measured the size of these cancer cells, and this is about what we would expect today. So I think a powerful recognition that these were scientists looking at this and wanted to quantify what they saw and actually measured things to basically what we would measure nowadays. And so right away, we have histologic features both in terms of language and in numbers. And this idea of morphometry, the quantification of these histologic features, really, exploded in the 1920s and beyond. And so we have in the 20s the first description of some things that I think are commonplace nowadays, that a cell doubles in size before it divides, and something I think we all recognize, that cancer nuclei are bigger than that of normal cells. And so these are like quantitative descriptions of these things. And probably like my favorite and maybe the seminal work from this era of morphometry has this wonderful figure. So this is Dr. Haumeder, apologies about my German pronunciations, who in 1934 published this paper looking at a quantitative description of the size of the nucleoli between cancer cells and non-cancerous cells. And I think, this figure amazes me that it's from the 1930s, I guess 90 years old this year. And I think with a little bit of cleanup wouldn't look out of place in a publication today in 2024. But, I think amazing generating these by hand, these distributions of the sizes of these this particular histologic feature, the size of the nucleolus. And this work was presented the year before at the annual meeting of the AACR, the American Association of Cancer Research, in a little blurb published in a little journal you may have heard of called Science. And I love this particular sentence. And this is a description of a presentation at the AACR annual meeting. They conclude that the cancer cell has at least one differential characteristic, and that this must be used by those who expect to reduce the mortality from cancer. And I think very interesting thinking back, this is 91 years ago, and today we've identified quite a few differential characteristics that have in fact reduced the mortality from cancer. Many of those with a molecular underpinning, so we can use targeted therapies, but I think also some histologic-based features that pathologists use nowadays to really understand what we're looking at. And, these histologic features, if we fast forward to today, are how pathologists learn to practice pathology. And so we have a quantification, like a combination rather, between some quantified histologic features. So this is all about gastrointestinal stromal tumors. And for that type of sarcoma, the mitotic rate and tumor size are very important to understand the behavior of a particular tumor. And so we have some quantified histologic features that deal with those. But then we also have, microscopic descriptions in natural language. And so what you do, or one of the things that pathologists do to learn the practice of pathology, is they open up a big textbook like this one, and they flip to a section and they read this whole paragraph, and they understand maybe a bit better after reading what they're expected to see when they look under the microscope. And we often have this combination here on the right, where we have like a couple regions of interest and a description in natural language of what we're looking at. And that's like basically how you learn and sometimes how you practice pathology. So often if you get a slide or from a tumor and you're not exactly sure what it is, you could flip to the appropriate section in the textbook and try to do some pattern matching between the images that you have captions for and what you're looking at under the microscope, and try to get closer to a diagnosis that way. And so I think, nowadays, I think if you have some histology images and you need to understand what's in them, you really want to extract some histologic features. a totally valid thing to do is to give those slides or those histology images to a pathologist, and they might look at low magnification, as on the left. This is another gastrointestinal stromal tumor, or GIST. You might look at high magnification, as on the right, and they could generate for you a microscopic description. And that microscopic description may have some quantified histologic features, but will mostly be in the sort of natural language, the jargon of pathology. And then, let's say you're doing this in a clinical perspective, and the pathologist may condense all that down into a final diagnosis and give, again, this combination of natural language. This is a gist, an epithelioid type gist, and then some quantified features, maybe some IHC and that sort of thing. And that's, I think, a totally valid way of extracting histologic features, and is certainly the way that's most important for patients today, even in 2024, is you have a human, a natural neural network interpret these images, extract some features, combine them into a final diagnosis, and that really drives the basis of all cancer care for every patient. But that's probably not why you've tuned into this talk, and I think we're probably all more interested in what can we do with AI or deep learning, and how can we, instead of having a very slow and expensive and subjective pathologist do this work of extracting features, can we do this in an automated fashion? And I'm sure that every person on this call understands that the answer to that is yes, but I want to talk a little bit about your different options there. So I think basically there have been two main paradigms of this feature extraction. We can extract human interpretable features, and so those are histologic features that, at least in theory, a human could look at and understand the basis for why that feature is what it is. And then some deep learning based features that likely have no human interpretation as possible for those features, but still, describe to a machine very precisely what's going on in that image. And let's talk about human interpretable features, that pathway first. And so these human interpretable features, as outside the context of using machines to do this, also has a rich history. And so this is, very probably the first work in computational pathology, maybe the second. How embarrassing to me, a pathologist, that the first work in computational pathology is from the Department of Radiology. I think it's shameful, but nonetheless, so this is Judith Pruitt and Mortimer Mendelsohn at University of Pennsylvania. And what they did was design this whole end-to-end system, essentially, with a microscope that can capture cell images hooked up to a general purpose computer. And this was back in 1966. So pretty amazing work. And, I guess even at that time, there had been other attempts to do this quantification of these human interpretable features, but it hadn't been possible until 1966. And if you go and look at this publication, there's some amazing images. And so here's a picture of a monocyte from that publication. This is 32 levels of grayscale all in one. And if you kind of squint or maybe move further back from your computer, or maybe if you're in the room and the screen is far enough away, that you can kind of get a sense for, some cytoplasm out here, and then some details within the nucleus here. And I love in 1966, the way they represented this 32 levels of grayscale is with the different characters here that their typewriter put out. So pretty incredible. But what they did was they hooked a microscope that captured this sort of image up to a computer that would then compute some histologic features for them. And so I think what exactly the features aren't super important. It has to do with optical density that's related to exactly what their microscope is doing. The microscope being called CyDAC. Forgive me, I forget what it stands for, but they were looking at whole blood cells, in this case, 4 different types of immune cells, and quantifying these features, and showed that depending on the human interpretable feature that you chose, you can separate these different, or the computer could separate these different types of immune cells based on that feature. And again, just a testament to the illustrative power that people had many, many years ago. There's probably my favorite image from this paper is this 3-dimensional recreation looking at cytoplasmic area, nuclear area, and this contrast optical density, and showed that, in this three-dimensional feature space, maybe like a proto-embedding space of these three different features, you can separate out these different immune cell types. And so, literally from the beginning of computational analysis of cell images, we have these human interpretable features cropping up. Nowadays, we have much more advanced ways of doing that, often with some deep learning baked in. And so, I've represented these as two different pathways, but often this human interpretable features pathway starts with some deep learning, at least today in 2024, to find particular parts of an image, and then we extract some features from that are human interpretable. And so I'm sure that everyone on the call is very familiar with probably the most important work in 2024 for extracting these human interpretable features, at least from an individual cell perspective. And so this is HoverNet, and I won't belabor it here, but the general idea is we apply some deep learning to a small region of interest, and that deep learning pipeline gives us a segmentation of all the nuclei in that image, as well as a classification of each nucleus. And so that classification will tell you a bit about the identity of the cell. And then the segmentation map that it produces, you can use that to calculate some human interpretable features like the area of the nucleus, the perimeter, all sorts of things. Kind of the world is your oyster once you have the segmentation of those nuclei. And one of the prototypical works that I always think about when someone says human interpretable features is this work from PathAI. And so PathAI, I'm sure I guess I'm not sure, probably does not use HoverNet to segment their nuclei, but instead they've paid, hundreds or thousands of pathologists to richly annotate pathology images, and then they train some deep learning models on those annotations to segment things like different cell types and different tissue types. And then once they have those segmentations, they extract all these, in this case, more than 600 different human interpretable features. And those could be, kind of crazy, like your fractal dimension of the cancer-associated stroma, to eccentricity of things, the area of the necrotic tissue, et cetera, et cetera. And in this work, they show that these human interpretable features actually are quite powerful at describing what's in these images, and that they have an association with different molecular alterations, which I think makes a lot of sense to pathologists. Pathologists can see, at least for certain tumor types, the differences in tumors that have, BRAF mutations versus non-BRAF mutations, that sort of thing. And so this is a nice version of that, and certainly goes beyond what a human could do. And then another kind of prototypical work, just as an example of what you can do with human interpretable features, is this one from Anant Matabushi's group, where they train a deep learning model to extract to find some predefined features, in this case mitoses, and the nuclei and tubules, and then show that once you aggregate those features together, you can do some feature selection and then show that these human interpretable features are actually prognostic, have some prognostic significance. And I think this is kind of, both this and the PathAI work, shows the kind of attraction of these human interpretable features, and that you can define them ahead of time. We know exactly what they mean, say the number or density of mitoses, and we can connect them either to some molecular alterations or some prognosis here. And then I think is very attractive to someone with a biological background who really wants to understand, if I have two tumors, one of which look kind of identical, but one, the patient survives for a very long time, and one for the patient does not survive for a very long time, wouldn't it be nice to use some, let's say, AI model to classify these images and then be able to interpret what's going on there in the context of biology, and really try to understand a bit more about the behavior of tumors in general, maybe not these specific ones. And so, if you're really excited about human interpretable features, there's a lot of different options for you to extract those of, Histomics toolkit. And please correct me if I'm wrong. It's the only one that I found, the only software package I found that will do both the, segmentation of nuclei and the extraction of histologic features. For most other things, you kind of have to define your own human interpretable features to do, which I think, if you're just getting started in the field, or maybe you're coming from outside of computational pathology and you just have a bunch of histologic images that you want to extract some features from, my recommendation is to start with the Histomics toolkit because things are predefined for you. But, as I said earlier, HoverNet is really kind of like the standard of care for human interpretable features, at least for segmenting nuclei. And so another good place to start would be start with HoverNet, segment your nuclei, and then predefine some histologic features and extract from there. And in the past couple months and years, we've seen other people trying to improve on HoverNet and other different methods of nuclear segmentation. And I have to call out HoverFast, which just got a preprint loaded from Andrew Janowick, who's part of the group at Emory, and claims some speed up over HoverNet. But we'll see. A lot of good options here. Maybe I'll pause there and ask for questions. I don't see any in the chat yet, but.
Do you have any questions in the group so far?
Just a quick comment. I guess one of the software that also extracts human interpretable features, UPath.
Yeah.
Oh, yeah.
Yeah, I didn't know Histologic PK does it, to be honest. I didn't know it does segment at all. I thought it's the annotation tool. It's good to know.
No,
They have updated it.
I suppose a quick question on the latest sort of methods that you put here, such as HoverNext and HoverFast. In terms of, have you tried them out in your lab? Have you seen what the performance is like? I know, of course, I think both HoverNext and HoverFast claim about a 20 times speed up, don't they? Which is fantastic if they can. So
yeah, I haven't personally tried out either of them, but I know, like the group I did my postdoc with, Faisal Mahmood's group in Boston, we've done some stuff with cell VIT, and it appears faster, but it's not like it's harder to do like a direct comparison.
yeah.
And looking at the preprint for HoverFast, and so I've talked to Andrew Janowick, who's helped develop HoverFast, and he's quite excited about it. I think in the preprint, the average dice score is like 0.91 or something, and as compared to HoverNet. But it's a little unclear to me what that means. Like, is that good enough? I'm not sure. Like, yeah, but a 21 times speed up or so. Yeah.
Yeah, exactly. So, amazing.
We'll see.
Yeah,
Other questions? Yeah. So most of the tools that have been mentioned here, they're about nuclear segmentation. But of course, when we talk about human interpretable features, lots of the time we're also interested in other objects in histology. So do you have any idea of if there's any public tool or a specific approach for detection of objects, like bigger objects, say glands? I mean, we are aware of like some specific gland segmentation algorithms, like Cerberus, but obviously it's very different. I guess what I'm trying to say is that nuclear segmentation, the good thing with it is that it's common in all cancer types, but as soon as you go to other histology primitives like glands or perhaps tumor types or whatever, it just varies significantly from one cancer to another cancer. So what's comment on this? Like, do you think are you aware of any tool to be able to give us this kind of segmentation of those objects? And do you think if it's even possible to do that?
Yeah, wonderful question. An important question too. To be honest, I'm not aware of anything that will do something like that on a pan-cancer level. I think, as of today, we're kind of stuck in a realm where we need to either find some data set, maybe publicly available, and train our own segmentation models for these things for a particular tumor type, or maybe non-tumor type. But going forward, I think there's other methods, and I'll talk about one later, that have maybe decent performance, whatever that means, if doing this kind of segmentation in a zero-shot method. So you train a bigger model on a bunch of data of like cancer and non-cancer histology images, a pure deep learning model, and then, apply it zero shot to something like that. I haven't seen anyone investigating that thoroughly to see like what is the actual performance over a wide variety of cancer types. But, at the end of the day, like a prostate gland is going to look very different than a cholangiocarcinoma gland is going to look very different than a lung adenocarcinoma gland. And I think it's just kind of difficult nowadays.
Yeah, it's true. It is true. But as a pathologist, sorry, a quick question. So as a pathologist, would you think, so it's just a naive question really, how plausible is it, or how possible is it for you as a pathologist to do that, say, focusing on cancer detection, or gland detection in different cancer types? Can you claim that based on the knowledge that you have gathered, like in 3 years, you can do this across 40 types of cancer? Then we might have a hope for AI to do that as well. Or would you think it's completely impossible for a single person or a single AI to learn all that?
I definitely think there's hope. I think my performance would probably be pretty good at segmenting glands across cancer types. I think that I kind of doubt we'll ever reach perfect performance. I mean, whatever that may mean, only because like glands, depending on the tumor type and how poorly differentiated it is, the glands can look absolutely nothing like glands, and especially, and this is an issue with a lot of the nuclear segmentation tools, is like the boundary between nuclei or glands is like really poorly defined, and there's in some cases without some like multiplex immunofluorescence or something that labels the cell cytoplasm specifically, like we just have no idea from the H&E like where one cell ends and another begins, and therefore like what the actual boundary of a gland may be, especially when they're abutting other glands or have like very complicated shapes. And so I think there's certainly hope to do this. What the performance will be, I think, is not clear, and maybe the appropriate approach for if you were to set out to do a problem like this would not be using only H&Es. It would probably be using some combination of H&E and either IHC with some like cytoplasmic label to help you really delineate where these structures are, or immunofluorescence or something else that can do a bit better than H&E for that.
Thank you. Thanks, Drew. And we've also got a couple of comments in the chat. So Brenda has said, the current comment is very useful. In day-to-day work, very few pathologists spend time at high power and interrogate nuclear features. 90% of my time is spent at low power, at 20x or 100x across nearly all samples. Useful for diagnostics and grading.
Yeah, totally agree. I mean, but one of the things you learn in pathology residency is that the high power is for low power pathologists, which I think is like really probably not a useful thing, but also that high power, and by which I mean high magnification, is like can get you into a lot of trouble where you see things that you can get stuck on, and then the low magnification or low power is really sort of where you need to do your work. But yeah.
Thanks, Drew. There's also been a couple of comments from Luca who said that medical segment anything models would be your best bet for, I think, for glands. I think that was with reference to. And also mentioned that Histologic TK is the segmentation is relatively rudimentary, but I've not really had much experience with that. So, one comment. Brilliant. Well, thank you very much, and please feel free to continue then, Drew.
Okay. Okay, thank you, Zoom. All right. All right, so I've talked about the human interpretable features. Let's move on to deep features, and this I think will be, probably entirely review for everyone listening in, so I won't belabor it. This is kind of the prototypical publication that I think of, thinking of this kind of like contemporary era of deep learning applied to pathology images, Coudray et al. In 2018, where, and I'm sure everyone here is aware of, this is kind of like the standard pipeline for deep learning applied to pathology images. We get some data set, we split it up into our training, validation, and test sets. We split our histology image up into little patches, and then or tiles, or whatever your preferred terminology is, and then feed them to a deep neural network that's probably been trained for some other purpose. In this case, trained for natural image classification. We truncate that deep neural network, before it makes the classification and extracts some number of deep learning-based features. Maybe it's 256, maybe it's 768, maybe it's 1024, who knows? And then you aggregate, you use some method of aggregation of the deep learning-based features and make a prediction for the whole slide. And this is like incredibly common nowadays. And maybe now in 2024, we're seeing, 10 papers a day that use this sort of exact same pipeline, just with a little bit of variation maybe on each of these different steps. And this is a publication from our group a couple of years ago that, trained a very general method of doing this, but the feature extraction step, is kind of the same. We take our little image patches, we apply, in this case, some pre-trained neural network to extract those deep learning-based features. And, really, I think the standard nowadays is that you train a deep neural network somewhere, somehow. Maybe in 2018, 2019, you used a model pre-trained on ImageNet. But nowadays, I think there's a couple of different options for pre-training entirely on histology images for maybe better downstream performance once you have these deep learning-based features that go into the rest of your model. And there's been this rise in pathology and outside of pathology and foundation models. And so our group in Boston is one of the many groups that's published a paper about foundation models for computational pathology. And here by foundation model, I mean model that you train, often using a large amount of varied data, and then you use it for not one downstream task, but a full variety of downstream tasks. Sort of forms the foundation that you can use for many things down the line. And so this is from our paper in Nature Medicine, and we call our model UNI, and it's pre-trained on 100,000 or so whole slide images to do this deep learning-based feature extraction. And in the paper, we show that the features that it extracts are very expressive for what's happening in those little image patches. And so again, unfortunately, these deep learning based features come from an individual patch, and then you have to, have some subjective method of combining all the individual patch features into aggregate them somehow to get a deep learning based feature for the whole slide. And so maybe for your human interpretable features, that feature for the whole slide might be like a distribution of the value of those features, something like that. And here you can have very complicated aggregation functions. And we show that our model is good and does better on the tasks that we chose than some other models. I think, kind of unfortunately, a lot of the foundation model preprints and publications nowadays don't make their models publicly available, or they're pre-trained on entirely private data sets and that sort of thing. And so it's hard to compare them to other things, but they're certainly out there. And I'm sped through this section, but I'm sure it's review for all of you. But, if you would like to try your hand using UNI, our foundation model, we have a GitHub with some Jupyter notebooks that explain how it works. And you can actually download the model weights and everything from Hugging Face. And so you can take that model and apply it to your own histology images and see what you can do with it. I would say, C-Transpath is probably the most popular option for foundation models or models that have been pre-trained on histology images to do this feature extraction. And those features that C-Transpath extracts, though not quite as good as the UNI features are still really good and good for many, many different tasks. The caveat here is that C-Transpath was pre-trained on TCGA, and whether it's good or bad, I've seen that some reviewers get very uncomfortable if you apply C-Transpath features to TCGA images and use that to say that your method, whatever you developed, is working well, because it's been pre-trained on that. It's pre-trained in a self-supervised manner. There's no labels or anything. And so I think you could make an argument that doesn't really matter, but it makes people uncomfortable anyways. UNI does not use the TCGA images at all, only for like testing, some downstream tasks, we use them. But if you want to apply your model, so you're doing some like molecular alteration prediction, and you want to use a TCGA as a test set or a validation set or something like that, I would recommend not using cTransPath. A Korean company called Lunit is another one that has a publicly available foundation model that you can go and check out. And actually, the paper that I should have included here was one from Microsoft Research that came out of Nature last week, I think, maybe the week before. That's also publicly available, models available on Hugging Face. And you can extract your deep learning-based features and aggregate them to your heart's delight. All right, so I've talked about these like two main paradigms for histologic feature extraction. You got your human interpretable features and your deep features, and there's definitely some trade-offs for when you are deciding to make a choice between those two different types of features. And your human interpretable features, you define them ahead of time. They have a clear meaning, and they're for that reason easier to connect to the underlying biology. And because you're defining them personally, you sort of injecting this purposeful bias towards what features you think are going to be important in these histology images for whatever downstream task that you're doing. But because you're defining those features, they're kind of more manual labor is required to extract these features. Histomics Toolkit is very rudimentary for this sort of thing and is like maybe the only, or the other than QPath, way to extract to define and extract these features. Whereas the deep learning based features, you can kind of just like plug and play, and you don't really need to worry about how many features to extract, which features to extract, that sort of thing. But because of that, and the black box nature of these deep learning models, they're really difficult to connect to biology. And a lot of people, like my postdoc group included, have made these heat maps that are based on these deep learning based features for whatever task we're doing. And I think the utility of those is kind of debatable. I think if all of you care about is performance, and I haven't seen this rigorously tested, but my feeling is that if all you care about is performance, the deep learning based features are going to be a little bit better than your human interpretable features. And maybe that's okay. Maybe your purpose here only really cares about performance, and I think that's potentially fine. There's many things in medicine that we don't or did not understand how they worked before we started applying them to patients. And so like penicillin is the classic example of, we had no idea what penicillin actually did. We just know that it killed bacteria and helped patients. And it was only years and years later where we figured out the mechanism of action of penicillin. So it was totally opaque and difficult to connect to biology. And yet we use it because the performance was very high for this thing in medicine. And I think there's an argument to be made for applying these deep learning models to things in clinical practice if we can show that the performance for whatever task is very high and rigorously validate and show that there's no bad things that it does. But that's difficult to do.
So may I ask a quick question, sorry, Drew?
Yeah,
so in your opinion then, so are human interpretable features redundant now that we've got such good foundation models, except for explainability then?
I'm sorry, cut out for a second. Can you just repeat what you said?
Yeah, sure. So except for like using human interpretable features for explainability of models, do you think then that that human interpretable features are redundant then now that we have foundation models?
Yeah, good question. I mean, I think the explainability, I think, is probably key here, and is more than just explaining how the model works. I think for me, they're very interesting because of the connection to biology, and maybe the explainability of your model first is kind of this first step that could unlock many other things. And if we find some human interpretable feature that is, our model is definitely using because we have these nicer explainability things, then maybe we can use that histologic feature to understand the biology of the tumor a bit better, and then to design a better therapy or a better diagnostic or something like that based on the importance of that feature in your model. And so like just explainability maybe is not how I would phrase it. I think the explainability kind of is maybe, at least in theory, like a key to unlock an understanding of the behavior of a disease that you cannot unlock with the deep learning based feature.
Sure. Thank you.
Okay, so there's our two main paradigms for feature extraction. But for me as a pathologist, I find both of these like deeply unsatisfying. The deep features, of course, no connection to the biology, but maybe they perform really well. The human interpretable features, despite being human interpretable, like this is not how I or any pathologist thinks of tumors. Like I've never once thought about the fractal dimension of the tumor area or what the exact nuclear area of the tumor cells are or the density of lymphocytes, something like that. Like that's not only like some of these features, especially like the entropy of things, the gray level co-occurrence matrices, that sort of stuff, in theory is human interpretable, but is not really like pathologist interpretable. And both of these things kind of throw away all the work that pathologists have done using our natural neural networks to understand histology images. And if we go back and look at, this microscopic description of that gastrointestinal stromal tumor I showed a while ago, there's all this pathology jargon that we use that actually have like very clear meanings to other pathologists and maybe to the treating clinicians about what's going on in the histology image that we're looking at, and that describes something about the behavior of this image. And so this is like, we've been using these natural neural networks for 130 plus years, looking at H&E stained images of cancer. And we've come up with this jargon that is very information dense and in conveying things to other pathologists about like what we're looking at, and therefore making a diagnosis and therefore like characterizing this disease. And this is one of my favorite pathology textbooks. I apologize, the font is rather small there, but this is from the Quick Reference Handbook for Surgical Pathologists. And there's like a whole chapter in this book that has these great tables that have a pathologist jargon like vesicular, what it actually means and where the term came from, and an example image for that histology feature. And so some of these features are individual cells, some like these two I'm showing here are more architectural features. And for me, this was kind of helpful because a lot of these pathology jargon terms originated back in like the 1910s and 20s and talk about, and were maybe in more commonplace use outside of pathology, but nowadays, like I've not used the word festoon or garland very often in my life. So it's helpful for me to see like a garland or a festoon. But this is like, again, like how we learn pathology and how we understand like what is in these pathology images. And, a common joke among pathologists that we love our food analogies. And, these are some of my favorite, these coffee bean-like nuclei that are nuclei that have little grooves in them, like this one here, maybe this one here, and these fried egg cells that are characteristic of oligodendroglioma. And what's interesting to me is that if you tell a pathologist this tumor in the thyroid has coffee bean-like nuclei, the pathologist knows immediately what the diagnosis almost certainly is. And then you have, just with this like 3-word phrase, have a very good understanding of how this patient's tumor is going to act, the natural history of this tumor, what the appropriate treatment should be, what are the things the pathologist should look like. So these are like incredibly information dense histologic features. And what I dream of as a pathologist is like these some kind of feature extraction that uses these terms that the literal blood, sweat, and tears of pathologists have gone into extracting by ourselves. And I think in the past 6 months or year, there's been some work that's kind of going along these lines. And so there's a paper from a group at Stanford led by James Sow, who has this visual language model that can extract some natural language features from pathology images. And then our group in Boston published a similar thing. And these papers are quite similar, very similar methods. I think the real difference is kind of where the data came from to train these. And they're both based on this this CLIP model from Facebook Research that uses this contrastive learning paradigm. And so the main idea here is that we are going to have an encoder for our natural language that is a feature extractor for natural language, feature extractor from histology images, And we want those features from these different modalities to live in the same embedding space, the same feature space, so that we can say, have like a correspondence between an image feature and a text feature. And basically the way you do that is you take your images and your text, you extract the same number of features, you put them in the same embedding space, and you say that, you know, if basically you create a data set that has pairs between images and text. And if the image and the text exist in the same pair, we want those features from both the text and the image to end up in the same spot in the embedding space. And if the image and the text don't correspond, then they end up in different spots. It's kind of a simple idea, but it turns out if you throw enough data at the problem, you can get very good performance from this sort of paradigm. And so on the left is an image from the Stanford Group's paper. And so they utilize medical Twitter, or maybe should be calling it medical X. If you're not aware, pathologists are very active on Twitter these days, often posting cases maybe as board review or interesting cases, or you'll sometimes get pathologists, you know, asking for help from other pathologists on Twitter, and say posting an image maybe with a microscopic description and saying, you know, what do you think this could be? Like, I'm stumped. It's a 30-year-old with this weird tumor. What do you think it could be? And then other pathologists will respond with some suggested diagnoses or IHC they could do, that sort of thing. And so you can get a very good, you know, very good pairs of images and text that way. Our group in Boston, we leveraged pathology textbooks that have those nice captions and images, and PubMed articles and that sort of thing. And, you know, both groups, we had to do a lot of like data cleaning here to get these, you know, perfect image and text correspondences. But once you do that, you can use the contrastive learning technique to get some pretty good models. And, you know, and this is just a representation of how it works at the end of the day. We do a cosine similarity between our image features and our text features, and that's why this embedding space is a circle. But you can do classification or retrieval in this way, where you encode a single image and a bunch of different options for text, and retrieve the text by just choosing the text that ends up closest to your image in that embedding space. And so you can do this kind of retrieval, or you could also kind of think of this as classification if you have you know, a finite number of different classes here. So this is that, if you're not aware, the lung adenocarcinomas have a couple of different architectural patterns that have some prognostic significance for the patients. And, you know, if you embed your text, say these different words, micropapillary, acinar, papillary, etc., and these images, and then you choose the word that ends up closest to that image in the embedding space, and classify or retrieve that. And it works pretty well. And you can do things with all kinds of histologic features, not just patterns of lung adenocarcinoma, but maybe normal features that are in those images, or necrosis, et cetera, et cetera. And, you know, what was kind of exciting to me was that you can use this in a generative fashion to kind of invert the method here and actually create captions for histology images. And here's some admittedly cherry-picked examples where this works really well. And you have a Hodgkin lymphoma. The top thing is what our model produced. The bottom is the ground truth from the pathology textbook, or something that I wrote, and it seems to work pretty well, at least on these images, that it picks out, you know, gets the diagnosis correct, and also maybe picks out some histologic features that I think are actually there. And what was cool to me was seeing some like error modes that this fell into. And so here's one where the diagnosis is wrong. So this is a cardiac rhabdomyoma here on the top left, but the model wants to call it a clear cell carcinoma or fat necrosis. So on the top right here is a clear cell carcinoma of the kidney. And the bottom here is fat necrosis. And if you squint, I think you can see a pretty good resemblance between these two and the cardiac rhabdomyoma. And so this is like an error that I'm sure that I made as a medical student or a beginning resident, and thought that was really interesting that the model was making this kind of like reasonable errors. Okay, so with this sort of thing, we're getting close to my dream here. We have these vision language models. We can insert a bunch of pathology images. We can extract some captions for those images the model makes, and maybe on top of that, you use a different model or maybe a GPT model or something like that to, clean up these captions into more structured text like I want. If you're excited about this, there's a couple options here, some publicly available ones. Conc is our model from Boston, Plip is the model from the Stanford group, and they're both publicly available. In our case, you can go to Hugging Face and sign up and get the model for yourself and extract these like natural language-based features. Again, to your heart's content. All right, so I was kind of lying to you before, like really there's kind of 3 different paradigms you can do here in 2024. You can extract your pathologist features, I'll call them, either with a pathologist or with a computer, your human interpretable features and your deep learning-based features. I see a question in the chat. Was the AI told the location of the biopsy for the cardiac tumor, or is it just reporting blind? Yeah, it was just reporting blind. So we just gave it this image and it produced this caption. So no other context based on the location of the image. So I think probably like a pretty difficult question if you were just to give that image to a pathologist, or maybe I think many practicing pathologists would be able to get that, but maybe a first-year, second-year resident with no other information other than the image would have a hard time with that. All right, there's the 3 different paradigms. I think, great question actually about context and the importance of context. One of the ways that you can give context to this sort of thing is a method I'll touch on very, very briefly. That we put a preprint out from my postdoc work last year that should hopefully will be coming out in publication form soon. And that's, hooking up this vision language model to a chatbot. And, wouldn't it be great to have a chat GPT, but that really understood pathology images and the language of pathology and medicine and that sort of thing. And very briefly, that's essentially what we did. The details about how we train this thing are in the preprint, and unfortunately, it's not publicly available yet, but hopefully when the paper comes out, we'll be able to make it publicly available for people to play around with. We evaluated this model and tried out a couple different things. The model's able to do these kind of multi-turn conversations that you would expect from ChatGPT, and so we give it an image and ask it to describe the tumor, and it can create a caption similar to what the previous model I was showing can do. And then you can ask it follow-up questions as well and say, here's an image of a high-grade serous carcinoma of the ovary, and it generates a pretty reasonable description and also gets the diagnosis correct and has an understanding of what IHC you should use for that. And similar, it's able to, in this cherry-picked example, like give a pretty reasonable grading for this prostate cancer. And then you can have, longer, more involved conversations of like having the model help you work up a case. And so, here's a left lung biopsy. What do you think this is? What IHC should I order? Okay, this IHC was positive or negative. What else would you recommend? And you can even go further, and I'm not showing it here, saying like, okay, like what molecular alterations would you expect, and what like treatment should I follow up with, and that sort of thing. And so I think it's not difficult to imagine that this sort of thing could act as like a help for pathologists, or maybe, in the AI and pathology world, I've heard people talk about this idea of a digital fellow. So when you're in pathology training, a lot of what your job is to get the slides, you write up the report, and you bring the slides and the report to your attending pathologist who looks at the slide, and in my case, crosses out big portions of the report that you've written and writes something entirely different. And then what they've edited goes into the medical record. And I think we all kind of recognize inside or outside of pathology that editing a report or text of any kind is much easier than creating it from scratch. And so I think, one of the ways this could be used is not have its results go into the medical record at all, but sort of prepare a first draft of the report for a pathologist to work on and edit. Or maybe, if we get high quality enough training data for rare tumors that pathologists in underserved areas don't commonly see, maybe a tool like this could help recommend some IHC or other details to look for to help them diagnose that case. And running short on time, very quickly say, evaluating this sort of model is very, very difficult. I mean, what we did was generate some like multiple choice questions and asked our model, GPT-4, and a couple publicly available vision language models these questions, and showed that our model did better than the others. The issue here is that, this data set is not publicly available. And so if we when we put it into GPT-4 or Copilot or Gemini or any of these like large language models or assistants or whatever that these large companies have built, then that data is going to be then part of the training data for the next iteration. So it's very difficult to have like a reproducible evaluation metric for this sort of thing, because the next time you ask GPT-4 these questions, it may or may not be doing better on them. Yeah, our model does pretty well, and yes, and so we also have some like open-ended version of this, which was difficult to evaluate. Had to get me and some other pathologists to like read the open-ended response to questions that, maybe we didn't provide it with these answer choices, just say, what's the most likely diagnosis? Or we ask it, what molecular alterations would you expect in this image, et cetera, et cetera. Yeah. So I'll leave you with, circling back to 1838 or 1840, these histologic features, I said everything new or everything old is new again. We're kind of reinventing the wheel here, where we can use machines now to extract these similar features that we were extracting by hand back in 1838, both natural language-based features and quantified features, human interpretable features as well. So I'll just leave you with this bit. This is by in the English translation, not the original German from that 1838 paper. We think that these observations will put an end to the confident anticipations expressed by some pathologists and entertained by many persons in that profession that some simple microscopic tests of malignant growths would be discovered. And so I think thankfully for patients and pathologists alike, we have discovered many simple microscopic tests for malignant growths. It turns out just having a human look at enough of these images, we can figure out what's cancer and what's not cancer and how these tumors are going to behave, et cetera, et cetera. But thought that was interesting. So yeah, I just started my lab here at Emory. If you're looking for your next step, please reach out to me. And thanks to everyone. Thanks to Adam for the invitation, and thanks for listening.
Thanks for a really fantastic talk. And I suppose with that, we'll open the floor to questions. Does anyone have any questions they'd like to ask? I know we've only got a couple of minutes before we need to finish up. So, can I ask a couple of questions? Yeah, questions?
That what I'm looking at people, if anyone else to do. All right. First of all, thank you for really great insight. So can you go back to the slide with the different paradigms of representing features? Yes, this one. So, so you mentioned that as a pathologist, what you really like to have some the features being the features that are being extracted, they be presented in the terminology that is more familiar to pathologists. So that made me think that maybe these computer or handcrafted feature, we call them, or which are human interpretable features as well, some of them might be easier to be translated into those pathologists like key features. For example, if you look for spindle cells, there are like some mathematical ways to measure how spindle-like a cell is.
Mm-hmm.
And that's interpretable. So would you think that still, because there are some methods that are doing that, and I still think yes, it's much easier to come up with some post-processing or even training models to convert these interpretable features into those pathologies acceptable feature names. I myself think that might be better because, well, first of all, those are interpretable, and because of the way they have been calculated, it's more controlled rather than black boxed deep learning models. But what do you think? Do you think there's some specific properties to these human interpretable features that still make them undesirable in comparison to this novel multimodal vision and language models?
Yeah, I think in theory, no, there are not. I think I'm a little skeptical of how able we will be to translate these human interpretable features into the like pathologist features, because as I'm sure you're aware of, many of these human interpretable features are either based just on like nuclear features or maybe based on the shape or something, or of like bigger regions of tissue that don't often capture the same things that I capture when I'm looking at them. For example, like the pattern of the type of stroma. Like we have this like desmoplastic stroma, which means it's like more it's usually more pink or maybe more gray because of the collagen deposition there. There's like more collagen in the area around the tumor cells sometimes than there is otherwise. And it's difficult for me to see, at least right away, like what human interpretable feature would capture that. Certainly some combination of them could, in theory, capture that, like maybe the color difference between this region of stroma and a region of normal stroma elsewhere, and maybe some like the length of the collagen fibrils or something like that. And so I think in theory it's possible to do the conversion that you're talking about. I think maybe today in 2024 with the data sets that we have been able to assemble, it's easier to do the kind of entirely black box thing and have more reasonable results. But, I think the ultimate dream would be to have the pathologist features be directly connected to quantified handcrafted features, human interpretable features, however you want to call them. I'm just a little skeptical like how difficult it would be and how general those features could be, if that makes sense.
Yeah, it'd be good if you could link up a path chat to these features, and then to throw out these specific features from path chat, say there's X amount of tills or
it's really interesting.
Yeah.
But like thinking of future of pathology, say imagine like 50 years from now, 100 years from now, and everything is digital, like whole slide images, and also it's easy to extract any kind of features from these images. Would you think there's still be a need for these sort of pathologists, pathologists, nomenclature for these image features? What I'm trying to say is that, for example, egg-like cells. Would you think that in 100 years from now, pathologists still need to describe the sample with those terminologies, or AI, because it will be more stronger than even like a couple of numbers from like an AI would be enough, would suffice? Or would you think now we would still need them for any reason?
Yeah, good question. Hard to conjecture. I think that at the end of the day, these pathologist features are subjective, and eliminating that subjectivity and doing that by having these quantified handcrafted features is probably where we will end up at some point. Yeah.
Okay. Thank you very much. Cheers.
Well, thank you very much. I'm very conscious that we're running about 5 minutes over, so I don't particularly want to take too much more of your time, Drew. So have we got any final questions, any final burning questions that anyone would like to ask before we finish the talk?
I see Brinder,
apologies
for pronunciation, has their hand up.
I think that's already been asked about. Is that a new question? 252. Was the AA told the location of the biopsy? I think that was.
Okay, address that. But if you have another question, then. And I'll just. Oh, I don't know what you're seeing right now. Hopefully not. I'll put my email in the chat. How about that? Or maybe Adam can send it out.
Brilliant. Oh, Buzz has got his hand up now. So have you got a question? Sorry, Buzz.
No, I suppose it's really to follow on from, I think, Mustafa's comments earlier. and I suppose Drew's as well. The idea of removing the subjectivity of all of these histological features it's really I think it's up for researchers like yourselves involved in this type of area to say, what is it that you want to develop? The truth is, in first world communities, digital pathology will become the norm, and we'll be doing more and more work on trying to correlate these features with the molecular outcomes and prognostics. In reality, you know, most of the world won't be digitized and still isn't digitized, but pathology is still required there. In truth, it will be whether or not teams like those here actually choose to want to remove subjectivity, and that will probably inform the future of AI. I mean, a very good example of this is, you know, the definition and diagnosis of phalloides tumors in breast, this idea of stromal expansion, and you know, lots of work can be done to try to define stromal expansion, and I think they've kind of now settled roughly on using 4 power fields, technically 40 regions of 40x magnification, and I actually really would be a proponent in saying we should be trying to look for converting this pathologist terminology and seeing if we can see if it actually does correlate correctly with prognostics or diagnostics, and if it does, trying to define it better. That would be in my I would argue that would probably be the best use of these tools, because that's likely to immediately transfer into the day-to-day working practices of every single pathologist. And there's only 2 other comments. There was idea of 100 years from now that will be all digitized. I don't think parts of the NHS are going to be digitized in 100 years from now. I'm still going to be writing down paper notes. And I suppose one element is any pathologist. This was a joke made to me by another pathologist who's now retired, when I was showing him papers much like the ones you described from like the early 1900s, where they're measuring nuclei and they're comparing the size of cytoplasm. I was told that this is only done by pathologists who hate their wives and children. So let's see. But anyway, thank you. Your talk is incredible.
Thank you.
Brilliant. Well, thank you very much, Drew. That was a really, really interesting talk, and yeah, so best of luck with the lab. I hope it goes well.
Thank you.
Yeah, so with that, I think we'll call the seminar to a close. Just a reminder that we've got our next talk in 2 weeks' time, so on the 24th, from 2 till 3, where we're joined by both Professor Stefan Frugel and Valentin Malinchuk from LMU Munich, Germany. But again, thank you very much, Drew, for a really interesting talk, and thanks everyone for joining in person and online.
Thank you all.
Bye, everyone. Bye.